The genetic technology that I wrote my research paper on was zinc finger nucleases, or ZFN for short. What is ZFN? To put it simply, zinc finger nucleases are enzymes that can cut DNA at a specified site. They're made up of essentially two things, a zinc finger and a half of an endonuclease called foci. The zinc fingers are made up of a chain of codons, which are usually 3 long, or 9 nucleotides in length per side. In total, there are normally 6 codons and a total of 18 nucleotides at each side of the strand of DNA. The zinc fingers are called that because there's a zinc ion in the center of the codon. This zinc ion contributes to the structure of the codon, which forms into a somewhat finger-like structure that you can see right here and right there. The picture on the right shows how each zinc finger is a codon long and also shows that there are normally 3 to a chain, as I mentioned before. So you can see right here that there's 1 zinc finger per codon. The other half of the zinc finger nuclease is the part that's attached to the end, the foci endonuclease. This was discovered when researchers were studying bacteria. They found out its function and took it for their own use. When combined with zinc fingers, the foci endonuclease forms into a full zinc finger nuclease that is able to cut the DNA, which here's a 3D rendered picture of what it would look like. This picture shows the process pretty well, I think. You can see the zinc fingers lined up along the strand of DNA from the 3' to 5'. Also, ZFNs are anti-parallel just like DNA. And on a side note, I found it interesting that this sort of looked similar to DNA replication. Anyway, now that I've described what each element is, we'll look at how it's done. The codons in the zinc finger chains are coded to target a specific part of the DNA. The longer the zinc chain, the more specific it is. When both sides of the zinc finger nucleases line up, the foci endonucleases bind together and cut the DNA in a sort of stair-step shape. That leaves a single line of nucleotide bases on each side, which I'll pull that picture back up. What you can see right here is that when they bind together, that it cuts in a sort of stair-step. The applications of using ZFNs are really promising for the future. Right now we haven't quite mastered using it to its fullest potential, but have been able to do some small studies verifying its capabilities. In the future, geneticists think it will be possible to use this to cut disease-causing genes out of our DNA. Some even think it could be used on cancer as well. Either way, this genetic editing technology has great potential in the future.